Walk into any old-growth forest, and you'll see what looks like competition. Tall trees racing for sunlight, roots crowding each other underground, a world built entirely on struggle, where only the strongest live. That picture is almost completely wrong. Beneath every step you take in a forest, there is a network. Not a metaphor, not a poetic image. A literal, physical, biological network made of living threads so thin that 100 of them bundled together wouldn't reach the width of a human hair. It moves nutrients. It carries chemical signals. It keeps trees alive that shouldn't be able to survive on their own. And it has been running continuously, without a single moment of downtime, for nearly 500 million years. Forests don't just grow, they are managed. And the managers are fungi. Not the mushrooms you see. Those are just the surface, the temporary fruit of something far larger and far older. The real organism is underground. It spreads for miles. In at least one documented case, it covers an area the size of a small city, and may be older than the Roman Empire. And right now, at this moment, while you're watching this video, it is routing resources, detecting damage, and making decisions about which trees live and which trees don't. Using a logic that took scientists most of the 20th century to even begin to understand. This is not science fiction. Every claim in what you're about to hear comes from peer-reviewed research, isotopic tracer studies, and field observations conducted over decades in some of the most intensively studied forests on Earth. The story is stranger than any fiction could make it, because the strangeness is real. If you haven't subscribed yet, this is a good moment to do it. What comes next is going to change the way you look at every tree you've ever walked past. The first thing to get right is what a mushroom actually is, because most people have it completely backwards. When you see a mushroom growing in a forest, you're looking at a reproductive organ. That's it. It's roughly equivalent to seeing a flower on a plant. It's the part whose job it is to make spores and spread them, and then it dies back. The flower is not the plant. The mushroom is not the fungus. The actual organism is called the mycelium. It's a web of individual threads called hyphae, each one a single cell wide, each one capable of extending, branching, and fusing with other hyphae to build something that has no fixed size, no fixed shape, and no obvious limit on how long it can live. Unlike almost everything else in biology, mycelium doesn't follow the rules that animals and plants follow. It doesn't have a body in any conventional sense. It's more like a process, a continuously growing, continuously branching, continuously dissolving and reforming network that responds to its environment in real time. Those threads move through soil the way water moves through a sponge, finding every gap, exploring every direction, pressing into spaces that roots can never reach. A single teaspoon of healthy forest soil contains enough fungal threads to stretch for several miles if you laid them end to end. Not several yards, several miles from one teaspoon. And this matters enormously, because the primary job of mycelium in a forest ecosystem is not to produce mushrooms. It's to be infrastructure. It's to connect things. For most of the 20th century, mycologists, scientists who study fungi, knew that fungi were in the soil. They knew fungi were associated with plant roots. What they severely underestimated was the scale. Early studies focused on single species, single host plants, small plots of land. The tools for seeing what was actually happening didn't yet exist. So the dominant model in ecology treated trees as fundamentally independent, competing for light, competing for water, each one an island, struggling to survive on its own. That model wasn't just incomplete. In important ways, it was the opposite of what was actually happening. In the early 1990s, a graduate student named Suzanne Simard was working in the old-growth forests of British Columbia. She had grown up in those forests. Her family had been loggers for generations, and she had watched the forestry industry clear-cut ancient stands and replant them with monocultures of Douglas fir, killing every other plant species in the process to eliminate competition. The replanted forests were struggling. Seedlings were dying at rates that the models couldn't explain. And Simard had a growing suspicion that the models were missing something fundamental. She designed an experiment that was, in retrospect, almost absurdly bold for a graduate student. She took birch trees and Douglas fir seedlings growing in forest plots. She covered the birch trees with bags of carbon dioxide, made with a special radioactive carbon isotope, carbon-14, so she could track exactly where those carbon atoms went after the trees absorbed them. Then she waited. The carbon didn't stay in the birch. It moved. It traveled underground, through the fungal network connecting the roots of the birch to the roots of the Douglas fir, and showed up in the tissues of the fir seedlings growing nearby. The trees were not competing, they were trading. The birch, sitting in sunlight with its leaves working at full capacity, was sending carbon sugars through the fungal network to the fir seedlings shaded beneath it, seedlings that couldn't photosynthesize enough on their own to survive. This was, to put it mildly, not what the textbooks said should happen. Simard submitted her findings to Nature, the most prestigious scientific journal in the world. The paper was rejected. She revised and resubmitted. It was rejected again. She revised once more, addressed every criticism, resubmitted. On the third attempt, in 1997, Nature published it on the cover. And the editors, not Simard, coined the phrase that would follow the research for decades afterward: the Wood Wide Web. The forestry industry's response was immediate and hostile. The implications of Simard's findings were deeply inconvenient for an industry built on the premise that forests are collections of individual trees that can be managed independently. If trees were connected into a network, if the health of a seedling depended on the health of the old-growth trees surrounding it, then clear-cut logging wasn't just ecologically disruptive, it was destroying infrastructure that took centuries to build. Industry-funded researchers challenged her methodology. Forestry policy in British Columbia didn't change. The pressure on Simard to soften her conclusions was, by her own account, sustained and professional. She didn't soften them. She spent the next 2 decades expanding the research. What eventually emerged from her lab, and from dozens of other research groups around the world, confirmed and dramatically extended the original finding. Carbon wasn't the only thing moving through these networks. Nitrogen, phosphorus, water, chemical defense signals, all of it was flowing through the fungal threads connecting trees across entire forest stands. The network wasn't a curiosity, it was the circulatory system of the forest. To understand how this works at the physical level, you need to picture something almost impossibly delicate. A fungal hypha, a single thread of mycelium, approaches a tree root. It doesn't just touch the surface and stop. Depending on the type of fungus, one of 2 things happens, and the difference matters. In the first type, called ectomycorrhizal fungi, the hyphae wrap around the outside of the root cells, forming a dense sheath, almost like a glove over the root tip, and then push between the outer root cells without actually entering them. The fungal threads weave into the spaces between cells, creating an interface where molecules can move in both directions. The tree gets phosphorus and nitrogen and water. The fungus gets sugars. In the second type, called arbuscular mycorrhizal fungi, the relationship is far more intimate. These hyphae don't just press against the root, they enter the root cells themselves. They push through the cell wall, and inside the living cell, they branch into a structure called an arbuscule, an intricate, tree-shaped structure that maximizes the surface area of contact between fungus and plant, where the actual molecular exchange happens. The fungal membrane and the plant cell membrane face each other across a gap measured in nanometers. 2 organisms, separated by less space than the thickness of a soap bubble, trading the molecules that keep each other alive. Arbuscular mycorrhizal fungi associate with roughly 80% of all land plant species. Ectomycorrhizal fungi are found mostly in temperate and boreal forests, the Douglas firs, oaks, beeches and pines that dominate huge portions of the northern hemisphere. In both cases, the deal is similar: carbon from the plant in exchange for nutrients the plant can't reach on its own. What makes this remarkable isn't the one-to-one relationship between a single fungus and a single plant. It's that the same fungal network can simultaneously connect dozens or hundreds of different plants, different species, different ages, growing in different light conditions, and route resources between them based on something that looks, from the outside, almost like demand management. A plant in deep shade that can't photosynthesize enough to feed itself properly becomes what ecologists call a carbon sink. It's pulling carbon from the network faster than it's contributing. A plant in full sunlight, running photosynthesis at maximum efficiency, becomes a source. The network moves carbon from sources to sinks, not in a trickle. In measurable, substantial quantities that can make the difference between a seedling surviving its first decade in a shaded understory or dying in it. And in 2025, researchers at Princeton and institutions in the Netherlands and France published a paper in Nature that finally revealed, in extraordinary detail, how the network manages this. They built a robot, custom designed for the purpose, that could track over 500,000 individual points in a fungal network simultaneously, following the trajectories of fluids moving through the hyphae. The network manages congestion by widening its channels near areas of high demand, near plant roots that are pulling hard, and narrowing them where demand is low. It's dynamic. It responds to conditions in real time. Inside each individual hypha, fluids don't move in one direction. They move in both directions simultaneously, in the same tube, like a two-lane highway packed into a thread thinner than a strand of spider silk. Nobody designed this system. It evolved over hundreds of millions of years of continuous operation, and it solves resource allocation problems that human infrastructure engineers still struggle to replicate in purpose-built networks. The oldest and largest known individual fungal organism on Earth is in Oregon's Malheur National Forest in the Blue Mountains. It's a species called Armillaria ostoyi, commonly known as honey fungus, though that name feels entirely inadequate for what it actually is. Researchers mapping it in the late 1990s and early 2000s used genetic sampling across the forest to determine which parts of the fungal network were genetically identical. The test for whether you're looking at one organism or many. A single genetic individual covered approximately 3.5 square miles of forest floor, nearly 9,000 acres of living mycelium, all connected, all part of the same organism. The age estimate, based on growth rate and area covered, runs somewhere between 2,000 and 8,000 years old, with the lower end considered more probable by most researchers. The more conservative figure puts its origin around the time the Roman Empire was at its height, and it's not the only one. Armillaria colonies of this scale have been found in Michigan, in Washington State, in Scandinavia. They are not rare curiosities. They are the natural endpoint of a growth strategy with no programmed stopping point. A strategy that produces organisms of theoretically unlimited size, with no maximum lifespan, quietly spreading through the soil across geological timescales. This is what a fungus actually is, once you let go of the cap and stem image. Not a small organism that produces mushrooms, a potentially vast, potentially ancient network that produces mushrooms the way your lungs produce breath, as a byproduct of a process that goes far deeper. And the network does much more than move nutrients from place to place. In a forest in Japan's Miyagi Prefecture, researchers inserted thin medical electrodes into 37 wild mushrooms growing from the same underground network and recorded their electrical activity every second for 3.5 days. When water was poured at the base of just one mushroom, that mushroom's electrical potential spiked, and within 30 minutes, the level of signaling across the entire network measurably increased. The whole system responded to a local event. More than that, one particular fungus emerged as a clear hub, sending significantly stronger electrical signals to the rest of the network, including to a mushroom of a completely different species growing at a distance. 2 different species, connected by physical threads of mycelium, exchanging electrical information in what the researchers described as a coordinated network dialogue. That result is still being processed by the field. The mechanism of cross-species electrical communication through fungal networks isn't fully understood yet. What's certain is that it happens. And back in the 1880s, a botanist named Bernhard Frank, working in Germany, tasked by the Prussian Ministry of Agriculture with figuring out how to cultivate truffles commercially, was the first scientist to look carefully at the fungal coating on tree roots and conclude, against fierce resistance from colleagues who saw all fungi as pathogens, that the coating was beneficial, that the fungus wasn't killing the tree, it was feeding it. He coined the word mycorrhiza, fungus root, in 1885. His insight was so far ahead of the field that his detailed obituary in 1901 didn't mention the mycorrhiza research at all. His colleagues apparently found it easier to praise his other work than to acknowledge what his most important discovery actually was. Science does this to people sometimes. The network, though, kept running, indifferent to whether any human understood it or not. What Simard found in the 1990s and what Princeton's imaging robot revealed in 2025 are just the most visible points on a much longer arc of understanding, an arc that keeps bending toward the same conclusion. The forest is not a collection of individual trees. It's a collective, an organism made of organisms, wired together by something that was old before the first tree ever grew. And that collective does things that start to feel, the more closely you look at them, genuinely difficult to explain with the usual language of passive biology. The network doesn't just move resources, it carries information. Information that triggers specific responses, changes specific behaviors, and alters the fate of specific organisms. Starting with the trees themselves, which have been sending messages through this network all along. The attack, when it comes, is not subtle. A bark beetle lands on a Douglas fir, somewhere in the Pacific Northwest. It bites through the outer bark and begins tunneling. The tree responds immediately, flooding the wound with resin, pushing chemical toxins into the damaged tissue. But the beetle is already inside, and it's not alone. Bark beetle attacks are coordinated events. Where one lands, hundreds follow within hours. The tree is fighting, but the math is against it. Now here's what happens next. In the forest around that tree, something changes. Neighboring trees, trees that haven't been touched, trees where no beetle has landed, trees that have no direct sensory contact with the attacked individual, begin producing defensive chemicals. Terpenes, tannins, compounds that make bark tissue harder to chew and more toxic to digest. They're preparing a defense against an attack they have no direct knowledge of. The question that consumed a generation of forest biologists was simple and almost impossible to answer. How? How does a tree know its neighbor is under attack before the attack reaches it? In the 1980s and early 90s, the leading theory was airborne volatile chemicals. The attacked tree releases compounds into the air that neighboring trees absorb through their leaves and interpret as a warning signal. That mechanism is real. It exists. But researchers kept finding something that the airborne model couldn't explain. The warning signal was reaching trees that were downwind, trees in locations where the airborne chemicals couldn't possibly have carried. Something else was moving the message. The underground route was proposed before anyone had the tools to prove it. The idea was treated with considerable skepticism. Plants were understood as passive organisms. The notion that they were actively routing warning signals through a shared biological network felt, to many mainstream ecologists of the time, like an overreach, a fantasy, the sort of thing that sounds appealing but doesn't survive contact with data. Then a researcher named David Johnson, working at the University of Aberdeen in Scotland, designed an experiment that made the data unavoidable. Johnson grew tomato plants in controlled conditions, some connected through mycelium, some deliberately disconnected by physical barriers in the soil that mycelium couldn't cross. He then infected one plant in each group with aphids, and he measured what happened to the other plants. The result was clean and replicable. In the connected groups, the unaffected plants began ramping up their chemical defenses within hours of the attack on their neighbor, before any airborne signal could have reached them, before any aphid had moved to their leaves. In the disconnected groups, nothing happened. The plants sat there, unaware, producing no defensive response at all. Johnson later said in an interview that when he first saw the data come back, his reaction wasn't excitement. It was suspicion. He went back through the experimental controls, looking for the error. He rechecked whether any of the physical barriers in the disconnected groups had been breached. He ran the experiment again. The same result came back. The signal was traveling through the mycelium. There was no other explanation that fit. The warning signal wasn't a simple chemical gradient diffusing through soil. It was a message of some complexity, a mix of compounds that the receiving plants could apparently interpret with enough precision to begin producing the specific defensive chemicals most relevant to the specific threat their neighbor was facing. Not a generic response, a targeted one. To picture how this works underground, drop down below the surface. The soil in a mature, temperate forest is not dirt in any sense that word usually implies. It's a living structure, a three-dimensional lattice of mineral particles, organic matter, water films, air pockets, root channels, and biological activity so dense that a single cubic centimeter contains millions of individual organisms. Through this space runs the mycelium, which at this scale looks less like threads and more like a pale, branching river delta seen from altitude, endlessly dividing, endlessly reconnecting, forming loops and redundant pathways that give the network resilience against damage. At the nodes where fungal hyphae meet plant roots, chemical molecules move both ways continuously. The plant loads sugars into the interface, glucose, fructose, the direct products of photosynthesis, and the fungus picks them up and routes them through its threads. In exchange, phosphorus atoms that were locked in mineral compounds the plant's roots couldn't chemically dissolve get mobilized by fungal enzymes and delivered directly into root cells. Nitrogen follows the same path. In phosphorus-poor soils, which covers a large fraction of the world's forested land, this exchange isn't a bonus, it's survival. Without the fungal partner, the plant cannot access enough phosphorus to grow. The dependency is that fundamental. But the exchange is not equal across the network, and it is not random. The network has a topology, a structure of connections, and some nodes in that structure carry vastly more traffic than others. In Simard's work in British Columbia, a pattern emerged that she pursued through years of follow-up research. The oldest, largest trees in a forest stand, the ones with the deepest root systems, the most extensive fungal connections, the longest history in that particular patch of ground, were consistently sending more resources into the network than they were receiving. She called them hub trees, and later, in a term that attracted both attention and criticism for its anthropomorphic implication, mother trees. Whatever you call them, the functional reality is this. A 200-year-old Douglas fir, sitting at the center of a dense fungal network, is actively subsidizing the growth of seedlings in the understory beneath it. Not all seedlings equally. The research that made this genuinely strange was published by Simard's group in 2015, and it addressed a question that most people wouldn't think to ask. Does the mother tree know which seedlings are its own offspring? The isotopic data suggested yes. When seedlings grown from the seeds of a specific mother tree were planted near that mother tree, genetically her offspring, she allocated significantly more carbon to them through the fungal network than to unrelated seedlings of the same species planted at the same distance under the same light conditions. Not a little more, substantially more. The difference was measurable, consistent across multiple experimental replications, and couldn't be explained by any physical variable other than genetic relatedness. How a tree recognizes its own offspring underground, and how that recognition signal is interpreted by a fungal network and translated into differential resource allocation, is a question without a complete molecular answer. The outcome, though, is established. The forest has hierarchies, preferences, and something that functions at the level of observable behavior, like favoritism. And now for the other side of that equation. The same network that nurtures seedlings can strangle competitors. Some trees, particularly in temperate forests, form tight partnerships with specific fungal species that are hostile to the fungal partners of other tree species. The fungal networks don't just trade, they compete. And in that competition, the network itself becomes a weapon. Black walnut trees have been known for centuries to kill plants growing near them. Gardeners have observed for generations that few things survive in the soil beneath a black walnut's canopy. The chemical responsible, juglone, was identified and studied extensively as an above-ground phenomenon. What's less discussed is that fungal networks connected to black walnut roots carry and concentrate juglone compounds in the soil in ways that extend the toxicity zone far beyond what the root system alone could achieve. The network amplifies the weapon. More broadly, research into the competitive dynamics of mycorrhizal networks has documented something that complicates the Wood Wide Web's cooperative narrative significantly. The same channels that carry resources from source to sink can be used by well-connected hub trees to suppress the growth of competitors, particularly competitors that would otherwise begin shading the hub tree's canopy or competing for its water supply. The mechanism involves both direct chemical suppression and something more indirect. The hub tree's fungal partners colonize soil that competing plants need for their own fungal associations, effectively denying competing plants the mycorrhizal connections they need to access phosphorus and nitrogen. The competitor doesn't get attacked directly, it just finds itself unable to form the partnerships it needs to survive, and slowly fails. The forest floor, which looks from above like a quiet, peaceful arrangement of trees in available space, is an arena of continuous underground competition, managed by networks that have been optimizing their strategies for millions of years. Species composition in a given patch of forest is not just a product of which seeds fell where and which got enough light to germinate. It's a product of which fungal networks were already there, which trees they were loyal to, and how effectively those networks suppressed incoming competition. Some patches of forest function underground, more like territories than like open ecosystems. And then there are the fungi that skip the subtle approach entirely. Ophiocordyceps unilateralis is a parasitic fungus that infects carpenter ants in tropical forests. The infection begins when a fungal spore lands on the ant's exoskeleton and germinates, pushing filaments through the cuticle into the ant's body cavity. The fungus begins consuming non-essential tissues, fat reserves, some muscle, while keeping the ant's critical systems running. It is, in a very real sense, farming the ant from the inside, managing resource consumption carefully enough to keep the host alive and mobile. Then, the behavior changes. The infected ant begins to leave its colony's normal territory. It descends from the canopy, where carpenter ants typically forage, and begins moving through the understory. It climbs vegetation, but not to any random height. Studies conducted in Thai tropical forests found that infected ants consistently ended up biting into plant tissue at a height of 25 centimeters above the soil surface. Not roughly 25 centimeters, consistently, precisely, within a range of centimeters across multiple forest sites and multiple ant colonies. The temperature and humidity at that specific microhabitat height are optimal for the fungus's subsequent reproductive stage. The ant bites into a leaf vein, using its jaw muscles to clamp down with a force that leaves a characteristic scar in the plant tissue, and dies. The fungus then completes its life cycle inside the dead ant, sending a fruiting stalk through the ant's head and releasing spores to rain down on the forest floor below, where other ants walk. For years, the assumption was that the fungus was hijacking the ant's brain, invading neural tissue and forcing commands. In 2017, a research team led by Maridel Fredrickson at Penn State used three-dimensional microscopy to map exactly where the fungal cells were inside infected ants at the moment of the death bite. The fungal cells were not in the brain. They had infiltrated the muscles of the legs and jaw, forming a network of cells between the muscle fibers, physically surrounding each muscle cell with fungal tissue. The fungus was not issuing commands to the ant's nervous system, it was controlling the muscles directly, while the ant's brain remained structurally intact, possibly still receiving signals from a body that was no longer responding to them. The ant may have been, in some sense, aware. We don't know whether ants experience anything like awareness, but the mechanism itself is what stops you. A fungus that evolved to control animal movement without touching the nervous system, operating instead at the level of individual muscle cells, with a precision of outcome that includes specific altitude, specific compass orientation, and specific biting behavior, all perfectly tuned to the fungus's own reproductive needs. Over millions of years of evolution, something developed that functions like a remote control for an animal's body, built from biology, operated by a fungus. That capability, interfacing with animal tissue at a deep level and altering behavior, doesn't appear only in tropical ants. It appears, in different forms and through entirely different mechanisms, across the fungal kingdom. Which brings us to a fact that most people have never been told about their own biology. Every healthy human gut contains a fungal community. Not bacteria, fungi, the mycobiome. It's made up primarily of species from the genus Candida, along with Saccharomyces, Malassezia, and a shifting population that varies significantly between individuals, depending on diet, geography, medication history, and dozens of other factors. Your lungs have a fungal community. Your skin has one. These are not the same organisms as Ophiocordyceps. The mechanisms at work are completely different, but the underlying principle is the same one running through every story in this video. Fungi and animal biology are not separate systems. They are entangled. For most of the history of medicine, this fungal layer was treated as irrelevant background, not pathogens, not important, not worth studying unless something went visibly wrong, like a candida overgrowth causing obvious symptoms. The assumption was that the fungi were just there, passive residents. That assumption is now being revised, and the revision matters. Research from the last decade, conducted at institutions including the Pasteur Institute in Paris and the University of California, San Diego, has established that the gut microbiome interacts dynamically with the bacterial community, with the immune system, and with the gut lining cells that form the barrier between the intestinal environment and the bloodstream. Disruptions to the fungal community, through antifungal medications, through dietary changes, through the collateral damage of antibacterial antibiotics that alter the competitive balance between bacteria and fungi, are associated with inflammatory bowel conditions, with increased intestinal permeability, and with systemic immune dysregulation. And then there's the gut-brain axis, the bidirectional communication pathway between the intestinal nervous system and the central nervous system. It operates through multiple channels: the vagus nerve, hormonal signaling, immune messengers, and metabolites produced by gut microorganisms. Bacterial residents of the gut influence the production of neurotransmitter precursors. The gut produces roughly 90% of the body's serotonin, but it's worth being precise here. Most of that serotonin is used locally, within the gut itself, to regulate intestinal movement and signaling. The blood-brain barrier prevents most gut serotonin from crossing directly into the brain. What the gut microbial community does, including its fungal members, is influence the precursor molecules and immune signals that feed into broader neurological regulation, a less direct, but still consequential pathway. Research from Ilyan Ilyev's group at Weill Cornell Medicine has shown that specific fungal species in the gut directly shape the immune signaling environment in ways that reach beyond the gut. In mouse models, disrupted gut microbiomes showed altered immune responses in the brain, changes in the function of microglia, the brain's resident immune cells. The bridge between fungal gut community and brain function is not a speculative idea. It's an active area of investigation, with preliminary results that are, to put it carefully, not reassuring about how little we currently understand. We are walking ecosystems. The fungi in your gut, on your skin, in the mucous membranes of your lungs, are not passengers. They are participants in processes that shape how your immune system fires and how your gut barrier holds up under stress. And if the current trajectory of research continues, they may turn out to be participants in something that goes deeper still, into how you feel, and how your brain processes the world around you. Which brings us to a compound that a fungus has been producing for millions of years, that humans encountered long before they had a name for it, and that neuroscience has only recently found the tools to study with any precision. In a research suite at Johns Hopkins University in Baltimore, a man in his mid-50s sits in a reclining chair with a blanket over his lap and an eye mask over his face. Two therapists sit quietly in the room with him. Soft orchestral music plays. The man is not asleep. He is, by clinical standards, in one of the most profound, altered states a human nervous system can enter without losing consciousness. He had been referred to the study through his oncologist. The diagnosis was stage 4 cancer. The clinical reason for his participation was treatment-resistant depression and existential anxiety, a condition so common in terminal patients that it has its own diagnostic category and so poorly served by existing treatments that Johns Hopkins received special DEA scheduling exemptions to study it. The compound he had been given 4 hours earlier was psilocybin, a molecule produced naturally by several dozen fungal species, synthesized here into a pharmaceutical capsule at a carefully measured dose, within the range used across the study's clinical trials. 4 hours in, he starts talking, quietly, in fragments, the way people talk when they're trying to describe something that language wasn't built for. He says he can feel the boundaries of himself becoming uncertain. Not frightening, uncertain in the way that a question you've been carrying for decades suddenly stops feeling urgent. He says that for the first time, he doesn't feel like a person watching the world from behind glass. He feels like he's inside it. Like the distinction between where he ends and where everything else begins is a convention he'd been following without realizing it was optional. Later, in the formal interview that is part of the study protocol, he says it more directly. I understood, in a way I've never been able to understand anything before, that I was not separate, that I had never been separate, that the idea of being a self contained inside a body, watching everything outside, that was the illusion. The other thing was the reality. The clinical team at Johns Hopkins had been collecting accounts like this for nearly 2 decades, thousands of participants, dozens of studies, and the accounts converged on something that proved genuinely difficult to explain with existing models of how the brain processes selfhood. Psilocybin, once it enters the body, is converted by liver enzymes into psilocin, which binds with extraordinary affinity to serotonin receptors, particularly the 5-HT2A receptor, densely concentrated in regions of the cortex most involved in complex cognition and self-referential thought. What happens next, documented in fMRI studies from Johns Hopkins, Imperial College London, and the University of Zurich, is a dramatic and consistent change in a specific neural network called the default mode network. The default mode network is the system that activates when you're not doing anything in particular, when you're daydreaming, remembering, planning, or thinking about yourself in relation to other people. Neuroscientists sometimes call it the network of self. It generates the continuous background narrative of being a person, the sense of continuity between the you of yesterday and the you of right now. Under psilocybin, this network's connectivity patterns change dramatically. The regions that normally talk only to each other begin cross-communicating with regions they almost never connect to. The rigid hierarchy that normally governs which information gets amplified and which gets suppressed becomes much flatter. The brain runs, temporarily, in a state of higher entropy. More signals from more places, competing without a clear arbiter deciding which ones mean something. The subjective experience of this is precisely what the man in the reclining chair described. The editorial voice in your head, the one that constantly frames every experience in terms of what it means for you specifically, goes quiet. Not silent, quiet enough that everything else gets louder. Other people's emotional states, the texture of music, the sense of being embedded in something larger than one person's continuous story. Roland Griffiths, who spent 40 years as a behavioral pharmacologist before turning his lab entirely to psilocybin research, said in one of his final public lectures that the most surprising thing about 2 decades of this work was not the clinical outcomes, though those were striking, with single sessions producing lasting reductions in depression and anxiety scores in a majority of terminal patients. The most surprising thing was how consistent the phenomenology was. Across cultures, ages, backgrounds, belief systems, the experience converged on the same core description: a dissolution of the boundary between self and world, a sense of fundamental connection, a lasting shift in how people understood their place in things. As a scientist who spent his career measuring and quantifying behavior, he said that level of consistency demands an explanation. The fungus that produces psilocybin didn't evolve it for us. The compound predates our species by a margin that makes the entire history of human civilization look like a rounding error. Genomic analysis, published in 2019, found that psilocybin biosynthesis evolved multiple independent times in different fungal lineages, not in a single ancestor that passed it down, but in parallel, on different continents, in different ecological contexts. In evolutionary biology, that pattern, called convergent evolution, typically signals strong selective pressure. Something about producing psilocybin was useful enough that evolution found its way to it independently, more than once. The most likely original function, deterrence of insect grazers, whose serotonin receptors are also disrupted by the compound, making the mushroom an unpleasant and disorienting meal. The fact that it also, incidentally, quiets the human sense of self and produces experiences of profound connection is what biologists call an exaptation, a feature that evolved for one purpose and turned out to have a radically different effect in a different context. Our serotonin receptors are close enough to those of insects that the same molecule hits us hard. We just have a cerebral cortex to process the impact in ways a beetle cannot. To understand what fungi actually are to this planet, not just what they do to forests or to brain chemistry, but what they fundamentally are in the history of life, you need to go much further back. In the late 1990s, a paleontologist named Carol Houghton was working with fossil material from the Devonian period, roughly 400 million years ago, and found herself looking at a problem that wouldn't resolve. The fossils were of a structure called prototaxites. Known since the 1850s, they had resisted definitive classification for 150 years. At their largest, these organisms reached heights of nearly 8 meters. Exceptional specimens, not typical ones. With trunks nearly a meter across, they dominated the landscapes of the early Devonian, the way tall trees dominate a forest today. Early botanists assumed they were plant-like. Then someone looked more carefully at the internal structure and found it was wrong for a plant. The tissue organization didn't match anything in the plant kingdom. When Houghton and her collaborators applied isotopic analysis to prototaxites fossils, measuring the ratio of carbon isotopes preserved in the tissue, the result placed it outside the plant kingdom without ambiguity. Plants fix carbon through photosynthesis, which produces a characteristic isotopic signature. Prototaxites had a completely different signature, one consistent with an organism that was heterotrophic, that got its carbon by consuming other organisms, combined with the internal microstructure, which showed interwoven filaments consistent with hyphal organization. The conclusion was unavoidable. Prototaxites was a fungus. Stand with Houghton in front of that realization. You're looking at a cross-section of fossil tissue under the microscope, and you're doing the math. 8 meters tall, built entirely from hyphal threads, at a time when the tallest plants on Earth were barely knee-high, before insects had evolved wings, before amphibians had crawled fully onto land. The largest living thing on the planet was a fungus, not a plant, not an animal, a fungus. The Devonian is sometimes called the Age of Fishes, because the oceans were full of them, and they get the dramatic press. What was happening on land during the Devonian was the single most consequential biological transition in the history of terrestrial life, and fungi were not bystanders to it. They were central to it. 450 million years ago, give or take, the first plants made it onto land. This transition required solving a problem that seems simple until you think about it carefully. Rock is not soil. The early land surface was essentially bare mineral. No organic matter, no structure, no nutrients in accessible form. Plants need phosphorus to build DNA and cell membranes. They need nitrogen to build proteins. On the early land surface, those elements were locked in silicate minerals that no plant root chemistry could dissolve. The fossil record makes this clear. The earliest land plants show mycorrhizal structures in their tissues from essentially the moment they appear. The fungi were there first, or arrived simultaneously. And the fungi brought a chemical toolkit that plants didn't have. The ability to secrete acids and enzymes that crack open mineral structures and released the phosphorus and nitrogen trapped inside. The plants provided sugars. The fungi provided access to the mineral substrate. The same deal that runs in every mycorrhizal relationship today, just the first time anyone had made it. A microbiologist working specifically on the genetics of arbuscular mycorrhizal fungi described the experience of studying this transition as reading the original contract. The genetic machinery governing the plant-fungus partnership in a modern oak tree is recognizably related to the machinery found in the earliest land plant fossils. It didn't need to be reinvented. It has been running, without interruption, across hundreds of millions of generations. A biological agreement made before insects existed, before dinosaurs existed, before the continents took their current shapes, still operating, right now, beneath every forest on Earth. Without that partnership, the land surfaces of Earth would have remained bare rock far longer than they did. The carbon cycle that controls Earth's atmospheric composition, the soil formation that allows agriculture, the forest ecosystems that regulate regional and global climate, all of it traces back to that first handshake between a photosynthetic organism and a fungus, made on a lifeless shore in the Ordovician. Now drive through a commercial timber plantation in the American Southeast, or a palm oil monoculture in Borneo, or any large-scale agricultural field treated with broad-spectrum fungicides to protect the crop. A forester who spent 2 decades moving between old-growth survey work and management consulting for timber companies described the transition between these two environments as something you feel before you understand it. In old-growth forest, the soil gives slightly underfoot. There's a sponginess, a give, because the fungal networks and the organic matter threaded through them create a 3-dimensional structure with air and water held within it. The smell is of active decomposition and growth simultaneously. You can smell the biology. In a clear-cut, replanted as a monoculture and treated with fungicides to remove competing vegetation, the soil is different in a way that registers before you've consciously analyzed it. Hard, compact. When you dig down, it's uniform. No structure, no layering, no hyphal threads visible at the root tips of the seedlings. The seedlings are growing. They have fertilizer and sunlight and water, but they are not plugged into anything. They are islands, struggling alone in a substrate that no longer has the infrastructure to connect them. The fungal network, once destroyed, does not quickly rebuild. In studies conducted in Oregon's forests following clear-cut logging, even 20 years after replanting, the diversity and complexity of the mycorrhizal community in replanted stands was significantly lower than in old-growth stands a few hundred meters away. The trees were growing in the way that a patient on intravenous nutrition is surviving, technically functional, but operating without something that had previously been doing enormous amounts of invisible work. The survival rate of seedlings in mycorrhizally depleted soil is measurably lower. Their response to drought stress is worse. Their ability to resist pathogen attack is reduced. Everything works less well. The economic logic of industrial forestry is to maximize wood volume per acre per year. The biological logic of a forest is to build resilience across centuries. These two logics are not compatible, and for most of the 20th century, the biological logic was simply not legible to the people making decisions, because the underground infrastructure was invisible, unmeasured, and therefore, in the accounting, worth nothing. The forester said it plainly: you can grow trees without the network, but you can't grow a forest. So come back now to the question running underneath all of this. We started with a forest that looked like competition and turned out to be a collaboration. We found that the collaboration is managed by an organism so old, so structurally different from anything in our intuitive model of life, that it barely fits into the categories we've built. We found that this organism doesn't just move resources, it maintains hierarchies, wages chemical warfare, and in some cases takes direct physical control of animal bodies with a precision that makes the word manipulation feel inadequate. And we found that a molecule produced by one branch of the fungal kingdom, when it reaches the human brain, does something specific and measurable and repeatable. It quiets the network responsible for the continuous story of being a separate self. And in its place, people consistently report something that sounds, across thousands of accounts from dozens of countries, almost identical. A felt sense of connection to everything around them. A dissolution of the border between inside and outside. The experience the man in Baltimore described, not imagining connection, but for a few hours, simply not filtering it out. I don't think that experience proves anything beyond neuroscience. What it reveals is something biological, that the default mode network, the self network, is a construction, a model the brain runs, and when it runs more quietly, what people encounter is the underlying reality that model was editing out, the continuous, physical, metabolic embeddedness of this body in the world around it. The man in the chair was not generating a hallucination. He was, briefly, perceiving something that has always been true. The oldest trees in any forest are not the strongest in isolation. They are the most connected. The ones that survive drought, pathogen attack, and resource scarcity are the ones embedded most deeply in the network, the most redundant connections, the most established partnerships, the longest history of exchange. Isolation in a forest is a slow death. The monoculture plantations make this visible in a way that's difficult to look away from once you understand it. Technically alive, biologically alone. Fragile in every way that actually matters when conditions get hard. The agreement that made all terrestrial life possible, struck on a bare rock shore before there were insects, before there were forests, before there was soil in any meaningful sense, was not a merger between equals fighting for dominance. It was an exchange, each organism offering what the other couldn't produce alone. That agreement has never been renegotiated, never been voided, never been replaced with something better. It is still running. In the roots beneath every tree you've ever stood under, in the soil your food grew from, in the microbial community in your gut right now. The fungi were here before us. They will be here after us, breaking down whatever we leave behind, feeding it back into the cycle, making the raw material available for whatever comes next. That's not a bleak thought, it's a clarifying one. The boundary between self and world is real enough in a practical sense. You end at your skin. But at the level of the processes that keep you alive, that built your body, that continue to maintain it, you are already in exchange with a world that assembled you from its own materials and expects, eventually, to have them back. The forest has known this for 500 million years. We're just starting to read the notes. If this changed the way you see the ground beneath your feet, subscribe. There's an entire world of science that looks nothing like what you were taught. And that's exactly what this channel is here for.